Hi everyone, so today we're going to be talking about Punnett squares and going over the basics of them to help you get through some problem sets or questions related to them. So first, let's just talk about genes. So DNA is this very long molecule that contains information that relates to proteins or regulation of proteins. And it's this double-stranded structure here, and it's wound around these proteins called histones that basically tightly pack it into a very, very tightly packed structure that we call chromosomes. And so chromosomes are various in thousands of genes that you can have in a person, and usually they encode for a protein or something along those lines. Now we could look at, say, like this chromosome here, and we could see, oh, it has this specific spot. This locus might have a specific gene that relates to specific phenotypes, so maybe hair color or height, or maybe if it's a non-functional gene, maybe it leads to a disease, like maybe it doesn't work, and then you have cystic fibrosis or things along those lines. So that's kind of, what genes are. They're segments along DNA. Now in genetics, we like to represent our genes and variations of genes with simple drawings and symbols. So I could draw out, DNA as a double strand and draw the nucleotides and try to link them together and say, oh, this region, represents a specific gene, gene A, perhaps. But that kind of takes a lot of time. That's kind of, us geneticists are a little lazy, so we might actually draw out, we could draw out the chromosome instead, here, the sister chromatids, and draw a centromere. I could label, okay, this region has, gene A. But that takes a lot of time too. So like I was saying, geneticists are a little lazy when it comes to drawing, so we like to draw it out with just simple either 2 lines or a single line, and we like to just label, okay, here we can find, gene A, perhaps. Now, the same thing goes with alleles. Now, alleles are variations of a gene, so you might see, a gene that's at a specific locus might have, a functional copy or a non-functional copy, and we write that out with symbols or letters. So we might say, earlier I was saying gene A up about here, so we might say, we have our allele A, and there's 2 variations, so maybe there's, allele A or big A, or maybe there's a version that's little a. So we sometimes can write with little prime symbols, so I might say A, put a prime, and A normal, and this, prime might refer to wild type being the normal, popular allele found in a population. So that's just kind of how we write out alleles. When we come to the topic of zygosity, that's when we're referring to, the relationship between different alleles. So an individual, say we have a person here, you've got 2 copies of every gene because you've got 46 chromosomes, 23 came from each parent separately. If they're heterozygous, that means you have 2 copies of each allele that are different for this gene. So you might say, A, little a, they have copy big A, and they have a second copy that is little a. Whereas a homozygous individual, might just have 2 copies of the same gene, and they are the same variation, so they're the same allele type. So we could write, big A, or, they could be something like little a, and there's any symbols that we decide for the specific gene or the disorder that we're talking about. So that's, the topic of zygosity. There are some other, terms like hemizygosity, which we may refer to, determining what type of chromosomes if you have 1 copy because of, non-duplicated types of chromosomes. But, here we're just going to talk about heterozygosity and homozygosity in the context of Punnett squares. So the next important topic that's important is gene dominance, and that's just referring to, when genes are being expressed and these variations between the expression. So, for example, we might have, gene A here, and let's say this gene A is a functional copy. And let's say, little a is non-functional. And let's say, you only need 1 copy. You only need, 1 copy is enough, which is called haplosufficient. We might say that, big A is dominant over little a. So that's how we'd write it, is we write with, a greater than or equal sign. And I might say, big A is dominant over little a, and that would be a dominant allele to the little a, which would be recessive. So dominance is a relationship between 2 genes. Now we're going to talk about crossing. So this is where we have, 2 individuals we're interested in, we want to predict and know what sort of offspring they might have. So if we have an individual that's big A, little a, and we cross them with another person that's big A, little a, we're questioning what proportion is their genotype going to be. Maybe it's big A, or big A, little a. We don't really know, and that's where we come into predicting and figuring out probabilities. So for predicting offspring, here's kind of a detailed walkthrough of what you could do and what's going on. So let's say, for example, we have an individual that has the alleles for this gene big A, little a, and we'll say that big A is dominant to little a. We might be able to, we could write them out. We'll say that this is individual 1 and this is individual 2. And if we were to write out the combinations, we could say, okay, they will donate, this person will donate one of the big A's, or they'll donate one of the little a's, and then individual 2 will donate, the same combination, either big A or little a, and then we're just going to see which could combine. So, we could have big A from individual 1 combining with big A from individual 2, which would give us big A, or, little a from individual 1 and little a from individual 2 to give us little a. We could have big A from individual 1 and little a from individual 2 to give us big A, little a, or little a from individual 1 and big A from individual 2 to give us big A, little a. So here we've got sort of a compilation of, you can see, it looks like 1¼ is big A, little a, 1/4 will be big A, little a, 1/4 will be big A, and 1/4 will be little a, little a. Or we could just write 1/2. So this is kind of a complicated way to do this, and this is where we bring in the concept of a Punnett square. So a Punnett square is taking that same, idea we just went through and trying to figure out what the offspring might be with a simple diagram. So if we had our individuals from earlier, big A, little a, we can just write them by filling in the firstmost column and the firstmost row. So for this individual, at the top, we might write in there big A, little a, the individual on the left will write there big A, little a, and then they're predicted, we combine the two. So we combine, big A, then we're going to combine the big A's here with the little a from this individual. And you can see we get that same, proportion of 1/2 were heterozygous, and then 1/4 were homozygous dominant or homozygous recessive for this gene and these alleles. So that's kind of how Punnett squares work. So let's put them to work. Let's see some examples, work through some problems. So here we have John and Sally are carriers. So carriers for a red hair allele, so they don't have red hair, they don't look like people that would have red hair. What proportion of children will have red hair? So I just have a pre-drawn Punnett square. This is the simplified way that I draw them. Let's draw out, their chromosomes that they'll have. And I'm going to write, our symbols as R, and we'll have the little r meaning red hair. So on one chromosome, John here has a big R and a little r because he's a carrier, and Sally over here also has one big R and little r. And now we will put these into the Punnett square. We'll have John on the left, we'll have Sally on the top. We'll get big R, little r, big R, little r. Now we can fill in our Punnett square. We can see, okay, what proportion of children will have red hair? Well, there's only one option where we will be homozygous recessive, and that is 1/4, r, little r, little r. So what proportion? 0.25 of children have red hair. Okay, so that's, making sense. Seems simple. Let's do another question. Okay, when crossing big A with little a, which offspring will be little a, little a? Okay, so let's just draw out our Punnett square here. I'm going to put big A, little a, and we'll fill it in. And you may notice, all of them are big A, little a. 100% are heterozygous. So what, which offspring will be little a, little a? The answer, none. So we would only be able to get little a if we took, the children of these two individuals, mated them, and the 1/4 of those would be little a, little a. So in this generation, not possible. Okay, here's another example. John's parents are both carriers for cystic fibrosis. His wife has no history of the disease. What are the chances his child will have cystic fibrosis, and what are the chances that John's child will be a carrier? Okay, so I've moved the questions over on the left, and we'll kind of just deal with this to start. So this might be a lot of information to try to keep track of, so I'm going to draw out a pedigree, which I went over in my previous video. So here we'll draw out John, and we will draw John's parents. And let's put our generation numbers, Roman numerals. John is 2-1. Let's draw out John's wife and their child, which is unspecified. We don't know anything about the child. Okay, so let's see if we can fill in the genotypes from what we know. So we know John's parents are both carriers of cystic fibrosis. So just to define our alleles, I'm going to write big F and little f. And you will see here that little f is the cystic fibrosis. Okay, so we know that John's parents are both carriers, so they must be heterozygous. We know that the wife has no history of cystic fibrosis, and it's a rare condition, and she's a founding parent for the generation 3 child, so it's very likely that she is homozygous dominant, so just a wild type, large F, large F. Now we need to figure out, okay, what is John, 2-1? Well, let's draw a pedigree, or not a pedigree, sorry, we just drew that. Let's draw a Punnett square of John. So I'm going to put both John's parents. Here we'll have large F, big F, little f. Let's fill it in. Very similar to what we just did. And that's a cursive F, if anyone's interested. A lot of times we write with cursive because sometimes lowercase might be difficult. So if I put if I was doing C as a letter for my symbols, large C and little c might be hard to distinguish if I just did that. So whenever we do lowercase, we often write in cursive, so I might put a tail right there just to indicate that I'm writing a lower c. So I highly recommend that you follow that pattern whenever you're trying to answer these questions to make it clear the difference between your alleles. Okay, so what are the chances? So here we'll look at John. Now you immediately might want to be thinking, oh, John, he has a quarter chance that he's, big F, 1/2 chance, large F, little f. But we know that John doesn't have cystic fibrosis because that isn't mentioned in the question. If he did, it would be significant enough for them to tell us. So we're going to actually remove this fourth category here with the little f, because we know he doesn't have that. And this is something called normalizing, which I can go into the math of it in another video, but here we're just going to look at it visually and see that if we look now, the proportions are that here only 1/3, because we have 3 boxes, it's large F, and 2/3 big F, little f. So if we're trying to figure out the chances of, their child, we need to have, John needs to be at least a carrier. Otherwise, if he has, big F, and he has a mating with his wife, which is big F, that means we'd be crossing big F with big F, and all of them would be 100%. So we know that's not the case in regards to what are the chances the child will have cystic fibrosis. Well, let's try if he's a carrier. I'll draw a Punnett square if he's a carrier, and I'll write out 2-1 for that individual. That's John. 2-2 is over here. This is the wife. And you can see, in order for their child to have cystic fibrosis, they need to be recessive homozygous, but that's not possible here. So there's no little f. Well, so what are the chances that their child will have cystic fibrosis? 0. 0 chance, because either he's homozygous dominant or he's heterozygous and can't, combine a little f with his wife, who we know is big F. The next question is, what are the chances that John's child will be a carrier? Well, if we look at the same situation that we were looking at earlier, we had a 1/3 chance John was big F, 2/3 chance John was big F, little f, and I'm just going to draw out that Punnett square again. If he was a carrier, then that means that half of his children will be a carrier as well. So you can see 1/2. Now this is under, and under something very important, and that's with him being a carrier, which is 2/3. So what we're going to do is we're going to take a product rule, which is and. So product rule, that's when you multiply two together. If it was or, then we would be summing. That's not important here. We're going to be multiplying. This is John, I'll write it out in writing, must be carrier, and child must be carrier. We would write out in math 2/3 multiplied, that's the and right there, by 1/2, which is the chances that they are a carrier, that's the second half. We get 2/6, which we can reduce to 1/3. That is the chance that John's child will be a carrier. Okay, thank you everyone for watching. I hope this helped. Don't forget to subscribe. That's really important to helping my channel grow, and suggest future videos, and hopefully you can do great on any exams coming up. All the best. Bye.